Bismillahirrahmanirrahim. Our today's lecture is Mechanism of Action of Antibacterial Drugs. I'm Dr. Amreen Ghani from Department of Chemistry, Bihari campus. These are the key concepts of the mechanism of action of antibiotics or antibacterial drugs. Introduction. The action of drugs and the sites at which they are believed to act are very varied. Common target sites are the cell envelopes, walls of microorganisms, enzymes, receptors, nucleic acids, and viruses. Now, what is the antibiotic or antibiotic drugs? Antibiotics are the substances produced by a microorganism or a similar product produced wholly. Means it could be synthetic or it could be partially semi-synthetic by chemical synthesis, that is capable in low concentrations of inhibiting the growth of bacteria or killing other microorganisms. These can be naturally occurring microbial products or any other synthetic agents that are used to treat the infectious diseases. Antibacterial antibiotics normally act by either making the plasma membrane of bacteria more permeable to essential ions and other small molecules by ionophoric action or by inhibiting cell wall synthesis. Mechanism of action of antibiotics. Actually, the antibacterial drugs can or antibacterial agents counter this attack by both bacteriostatic or bactericidal action. Bacteriostatic action occurs when the drugs inhibit growth of bacteria, hosts immune responses necessary to eradicate bacteria, with the result that it dies out naturally. Actually, in bacteriostatic, the antibiotics or antibacterial drugs, they somehow by the mechanism of inhibiting the growth of the bacteria or they inhibit the reproduction of the bacterial cells in bacteriostatic process, that its growth is stopped and automatically the bacteria stops growing and the antibiotic action occurs. While in case of bactericidal mechanism, in this the bacteria kills the the antibiotic kills the susceptible bacteria. The suffix static and side or sidal are widely used in other contexts to indicate these general types of action. Bacteriostatic antibiotics. These are the names of some drugs which are marketed as antibiotics whose action is bacteriostatics. These are tetracyclines, spectinomycin, sulfonamides, macrolides, chloramphenicol, trimethoprim. We will discuss their mode of action in the next slides. Now, bactericidal antibiotics, these are the penicillins, cephalosporins, fluoroquinolones, which include the ciprofloxacin, glycopeptides, vancomycin, its brand name, monobactams, and carbapenems. We will discuss in detail their mode of action in the next slides. Mode of action of antibiotics. Antibiotics act by disrupting various molecular targets within bacteria and cell surface, preventing the growth of the bacteria or initiating the killing of the bacteria. In general, drugs acting on microorganisms by either disrupting the structures of membranes and walls of that microorganism or their synthesis appear to act by number 1. Inhibiting the action of enzymes or other substances in the cell membrane involved in the production of compounds necessary for maintaining the integrity of the cell membrane. Actually, these antibiotic drugs inhibit the enzymes or some substances in the cell membrane uh, in order to rupture the cell membrane or to do inactivate the cell membrane. That's why the microorganism get inactive and cannot cause any bacterial activity. Number 2 is inhibiting processes involved in the formation of cell wall, resulting in an incomplete cell wall, which leads to loss of vital cellular material and subsequent death of cell. This is again the same point that the antibiotic drugs rupture or damage the cell wall or cell membrane so that the growth of the microorganism cannot proceed, it cannot grow, and its activity will be diminished. Number 3, forming channels through cell wall or membrane, making it more porous, which also results in the loss of vital cellular material and the death of the cell, and making the cell more porous by breaking down sections of the membrane. This is all about the action of the antibiotics. What happens actually is that the structure of cytoplasmic membrane is complex. Built into the membranes are the receptors and enzymes actually within the cell membrane or the cell membrane, and numerous channels connecting the intracellular fluid with the extracellular fluid. There are so many enzymes and receptors are present which interconnect the extracellular fluid through the ion channels. These channels allow ions and other small molecules to travel from the interior of the cell to its exterior and vice versa. Those whose primary function is the transport of ions are known as ion channels. Actually, the antibiotics block these ion channels so that the extracellular and intracellular fluid cannot be interconnected. Most drugs act on the receptors and enzymes found in the cell envelopes. When these cell envelopes will be disrupted or damaged, then how can the bacteria or microorganisms can damage the living organisms? However, a number of drugs act by either disrupting the structure of the cell membrane and walls or inhibiting the formation of cell membrane and walls or blocking the ion channels. When the cell membrane or cell wall is damaged or when the ion channels are blocked, then the activity of that bacteria or microorganism will be stopped. That's why how in this way the antibiotics show its mode of action. So, concluding on the previous slides, there are three broad mechanisms. One is the disruption or disrupt of the bacterial cell envelope, as I have told you in the previous slides. that disrupting the structure of the cell membranes and walls. Second, block production of new proteins. Third one is the inhibit the DNA replication of the bacterial cells. By blocking the production of the proteins or by inhibiting or blocking the production of DNA replication, the antibiotics or antibacterial drugs can stop the bacterial growth, or it can kill the bacteria in order to show the antibiotic action or antibacterial results. Let's go through these action that is performed by these antibiotics to prevent or kill bacterial cells. So let's begin with the synthesis of peptidoglycan or cell wall. This is the first antibacterial action of the antibiotics. This involves the synthesis of the peptidoglycan or the cell wall. These polypeptides, bacillin, beta-lactams and glycopeptides, which is vancomycin. These will disrupt the synthesis of the cell wall of the bacteria. Second is the cell membrane disruption. In second cell membrane disruption, the cell membrane bacterial cell will disrupt the synthesis of the cell membrane, thus causing the inner fluid to come outside of the cell along with the ions, etc., causing the osmotic lysis of the bacterial cell. Next is the destruction of the metabolic pathway of that produces the folic acids. And fourth one is the protein synthesis. Next is the RNA replication. If the protein synthesis stops, then the cell will not grow, and next is the DNA replication. First of all, we will see the peptidoglycan synthesis or the cell wall. For this, we will study the structure of the bacterial cell wall. The cell walls of all bacteria are being continuously replaced because they are continuously being broken down by enzymes in the extracellular fluid. Antibacterial agents, commonly referred to as antibiotics, often act by inhibiting their replacement biosynthesis of cell wall at any stage in its formation. Most drugs that act in this fashion exhibit either the formation of the precursor, starting compounds, the formation of peptidoglycan chains, or the cross-linking of the peptidoglycan. However, increasing number of bacteria are resistant to antibiotics, especially to beta-lactam antibiotics. This resistance is likely to become a major problem in future. Bacterial cell wall comprises of a rigid peptidoglycan layer having alternating amino sugars NAG and NAM cross-linked by peptide chains. The transpeptidase enzyme causes the transpeptidation in the final stage in cross-linking of the linear glycan chains. If when the antibacterial drugs or antibiotics stop the cross-linking of these peptidoglycan chains by removing the peptide cross-links of aniline glutamate, then the cell wall will be weakened and it cannot grow well, so the bacteria cannot grow in it.
The cell wall of gram-positive bacteria is composed mainly of thick layer of peptidoglycan. whereas the more stubborn gram-negative bacteria have an extra outer layer above the peptidoglycan layer. Peptidoglycans are composed of chains of amino sugars, such as NAMs or NAGs, that are cross-linked together by the short amino acid chains, such as alanine and glutamic acids. These amino acids are cross-linked together by the transpeptidase enzyme. To start their bacterial destructive effects, the beta-lactam antibiotics such as penicillin and cephalosporins have to pass through special barrel-shaped proteins known as porins that cross the outer layer of gram-negative bacteria. Then, penicillin must traverse the peptidoglycan and bind to specific proteins on external part of bacterial cell membrane called penicillin binding proteins or PBP. Binding to penicillin binding proteins activates penicillin antibiotics. Next, the active penicillin swims back to bind and inactivate the transpeptidase enzymes. The inactivation of transpeptidase enzyme results in the collapse of amino acid chains and damage of the peptidoglycan architecture. This leads to a series of reactions which end by massive destruction of cell wall and death of the bacteria. There are three major mechanisms for bacterial resistance to beta-lactam's antibiotics. Firstly, many bacteria have developed beta-lactamase enzymes that specifically bind to and inactivate the beta-lactam ring of these antibiotics. Therefore, beta-lactam's antibiotics become constrained and cannot attack the bacterial peptidoglycans. Secondly, some gram-negative bacteria close the porins and prevent antibiotics from reaching the peptidoglycans. Remember that there is no outer layer in the wall of the gram-positive bacteria. Finally, some bacteria develop mutations in the binding sites of the penicillin binding proteins, so penicillin is prevented from binding to them and remains inactive.
Antibiotics that inhibit the peptidoglycan synthesis. These are the antibacterial classes, polypeptides which include the bacitracin, beta-lactams and glycopeptides which contains vancomycin and teicoplanin. In this, the bacitracin disrupts the cell wall synthesis by the or the peptidoglycan synthesis, while beta-lactams include cephalosporin, penicillins, monobactams, and carbapenem. In this beta-lactam, the cephalosporin is a narrow spectrum antibiotic which includes only the gram-negative bacteria, while the penicillin is also the narrow spectrum antibacterial agent which includes the gram-negative bacteria, but the chemist has modified it to include the gram-positive bacteria also. While monocarbamates is a broad spectrum antimicrobial which includes both the gram negative and gram positive bacteria, while carbapenem is also the narrow range antibacterial agent which contains only the gram positive bacteria. These are the four classes of beta-lactams which contains cephalosporins, penicillins, monobactams and carbapenem. All these have different structures, but the main core of the structure beta-lactam, that is same in all these antibacterial agents. Beta-lactams bind to the penicillin binding proteins. These antibiotic drugs, when these bind to the protein binding penicillin binding proteins or PBP, they prevent PBPs from removing the NAM's terminal D-alanine. This in the last, when this D-alanine is removed from the NAM, then it prevents the cross-linking of the polypeptide strands. When cross-linking does not occur, the polypeptide chain gets weakened and the cell wall becomes weakened, and the bacterial growth cannot occur. Here in pink color, the beta lactams are shown. These will bind to the transpeptidase, bind to the PPB's transpeptidase. This is an enzyme which causes the cross-linking of the polypeptide chains. When the beta lactams binds to the penicillin binding proteins or PBBs, this transpeptidase cannot cause the cross-linking between the polypeptide chains, due to which the bacterial cell wall cannot grow. While in case of glycopeptides, in which vancomycin is present, this vancomycin will also bind to the D. This vancomycin will also bind to the D-alanine, and it will also not cause the cross-linking between the polypeptide chain, which leads to the eruption of the bacterial cell wall.
And that is the disruption of the cell membrane. This group of antibacterial drugs includes the polymyxins and daptomycin. Let's start with polymyxins. Polymyxins are polypeptides with lipophilic properties. They can insert themselves inside the lipopolysaccharide component of the outer membrane of the gram-negative bacteria, thus disrupting the outer membrane layer. Remember, this is the outermost layer of a gram-negative bacterial cell wall. They will then migrate to the cell membrane and repeat the process, killing the bacterial cell. Polymyxins target specifically gram-negative bacteria. They'll have very low selective toxicity, but they can damage the host kidney cells and nerve cells. Thus, the drug is only given topically as an ointment in the form of neosporin. Next is daptomycin. Daptomycin is a lipopeptide that works like polymyxin, inserting itself inside the cell membrane of gram-positive bacteria. The third mode of action is to prevent folic acid metabolism. Now, bacteria need to make folic acid. Unlike us, we get folic acid from our diet. However, bacteria cannot take folic acid from their environment. They must make it. Folic acid is important because it's a precursor to making adenine and thymine. These are two nucleic acids that help make up DNA. Thus, DNA synthesis is prevented if the bacteria cannot make its folic acid. In order to understand this, let's go through the folic acid pathway. It's going to be a multi-step pathway. First, para-aminobenzoic acid, also known as PABA, needs to combine with pteridine and L-glutamate to form dihydrofolic acid. Then dihydrofolic acid reacts with NADPH to form tetrahydrofolic acid, which acts as a precursor for DNA, RNA, and protein molecules. The antibacterial drug sulfamethoxazole is a member of the sulfonamide drugs that is very similar to PABA. Thus, it can compete with PABA to bind with pteridine. If sulfamethoxazole binds to pteridine, dihydrofolic acid is formed, and the rest of the pathway is stopped. Trimethoprim is another antibacterial drug that inhibits this pathway, but it looks similar to hydrofolic acid, and it will compete for its binding to NADPH. In effect, tetrahydrofolic acid isn't made, and sulfonamides and trimethoprim alone will decrease the production of folic acid to a bacteriostatic level, thus inhibiting bacterial growth, and are both broad spectrum. But if they're taken together, they can decrease the production of folic acid to the bactericidal level. This is a good example of antibacterial synergy. The next mode of action involves a class of drugs that inhibit ribosome function. Now remember, ribosomes help translate messenger RNA information to make proteins. Bacteria need proteins in order to live. Within this class of drugs, some will target either the small or the large subunit of the ribosomes. In each case, this effect will prevent protein synthesis. These drugs tend to be bacteriostatic and prevent bacterial growth without actively killing the bacteria. Just to remind you, the cytoplasmic ribosomes found in eukaryotic cells, such as ours, they are known as the ADS ribosomes. They're going to have similar functions of the ribosomes found in bacteria, the 7Es, but structurally different proteins. This will make for inhibition of protein synthesis a very good mode of action to treat bacterial infections. The classes of antibacterial drugs in this group will include the aminoglycosides, tetracyclines, macrolides, and chloramphenicol, to name a few. The aminoglycosides will target the 30S subunit of the 70S ribosomes. That's the small subunit. In effect, they prevent proofreading, which promotes the incorrect placement of amino acids, resulting in mutations at the protein level. Tetracyclines also bind to the 30S subunit, but at a slightly different place, preventing the incoming transfer RNAs from bringing in their amino acids, thus preventing the protein synthesis. Macrolides and chloramphenicol, on the other hand, are going to bind to the larger of the two subunits, the 50S subunit, and will prevent the formation of peptide bonds between the growing amino acid chain and the incoming amino acid. Like tetracyclines, both macrolides and chloramphenikols will stop protein synthesis. Where aminoglycosides causes mutations to the growing peptides, the effect is bactericidal, while the other three classes stop further proteins from being made, and this effect is felt as bacteriostatic. All four classes have broad spectrum activity on bacteria. The fifth mode of action is to prevent RNA replication, and the target in this case is RNA polymerase. The antibacterial agents that do this are known as rifamycins. They bind to the beta subunit of the initiating transcription complex and prevents the function of RNA polymerase. If RNA polymerase cannot transcribe messenger RNA from DNA, then further protein synthesis will also be prevented. The last mode of action is that of DNA replication, and the target here is DNA gyrase. Helicase is an enzyme that unwinds double-stranded DNA during DNA replication, which in effect will lead to supercoiling, as shown here on the right. In order for supercoiling for the DNA to be relaxed, DNA gyrase needs to unwind the supercoils to allow helicase to continue to open up the double-stranded DNA and thus continue DNA replication. Eukaryotic cells have a molecule similar to DNA gyrase. It's called topoisomerase. But because they are different proteins, quinolones and fluoroquinolones specifically target DNA gyrase and prevents it from functioning. This covers the 6 modes of action that antibacterial agents use to kill or inhibit bacterial growth, and thus concludes this video.